Alrighty, so part 2 of DNA replication. All right, so we'll start the second part of that. Before we start that, let's go back over a couple of things. What is a nucleotide? What are the 3 parts to a nucleotide? Phosphate. Okay. And what is the name of that 5 sugar or 5 carbon sugar that's in DNA? Deoxyribose. Good. And if it's an RNA, it's ribose. All right, great. So we have those two different kinds. What are my bases for DNA? Which one? Thymine is one, yes. Guanine. Cytosine. Adenine. So what is uracil? Where is uracil? Is it in DNA? It's in RNA. So if this was RNA bases, it would have all of these, right? But instead of thymine, it would have uracil. Everybody good? All right, what does Chargaff say? Chargaff's rules for base pairing. Adenine always bonds to or uracil if it's RNA, and then guanine always binds to cytosine. Good, good, good. All right, now I should be able to do this and tell you this is 5' and this is the 3' end. Can you name the complementary DNA strand? What would the complementary DNA strand be? Okay. Good. Now, if that's the complementary strand, what is the sequence of bases in their correct order? Okay, so we always read from 5' to 3'. So what end is this side on the complementary side? Has to be 3', remember? So this side has to be, what do we call it when they're going in opposite directions but they're parallel? Starts with an A. We could play Pop Test Hangman.
Okay. Oh.
I'm going to run out of room. Now I'm trying to spell it in my head. Okay, fine. You got those parts right. We're not going to play Hangman. Yeah, you got these down here. Because I said the word parallel in here and I realized I wouldn't have enough room. So, anti-parallel, right? Parallel because they go parallel. They're not going in the same direction. That's the anti part. But they're going next to each other. They're not perpendicular. They're like this. Okay? So,
Can we say the structure is anti-parallel, like we have anti-parallel structure?
Right. The structure is, yes, so you can say the structure is anti-parallel. What else about the structure do you know? Double helix. So if you haven't been doing the reading on this stuff, make sure you get the reading done. Okay? So there should be a 9.2 on our reading thing. You've got that linked in Canvas for you to do. So make sure you're reading it. And again, SQ3R is the best way to read a text in a life science and a social science. Okay? All right, so if that's the case, and we read from 5 to 3, what's the actual sequence, the correct order for the sequence for the complementary strand? A, G, right. So we're reading from 5 to 3. And we're reading from 5 to 3. That's how we read the sequence. And this will be important in protein synthesis. All right, everybody good? Okay, good. Well, we're all kind of caught up to speed here. All right. So let's grab our handy dandy little manipulative thing that we were working on the last time. And let's get us up to where we stopped. What's our first step in DNA replication?
Unwinding DNA strand out by filling hydrogen bonds.
Unwinding the DNA. Yes, you got it perfect. Unwinding the DNA. And then what's the enzyme? Helicase cuts hydrogen bonds. So DNA replication is a process, just like the processes we've been using. So you've got to make sure when you study, take a sheet of paper and then write down the steps and then take your notes and check it. You've just got to flat memorize it. Okay? All right, so we did that. We cut it. What do we use to cut it with? What did we use to cut with? What did we use physically? Scissors. So those scissors were our helicase, right? All right. Step 2. DNA polymerase. Yep. DNA. You're right. DNA polymerase adds. What does it add? Nucleotides. And it adds it very specifically. Yes, sir.
Do I actually cut like the?
Yeah. Don't cut these guys off. Yeah, you're good. If you got it cut and it just looks like this, you're right where you need to be. Yeah, you're great. All right, so adds nucleotides. What else do we know about DNA polymerase? To the 3' end only. Remember, it can only add it to the 3' end. So far, so good? All right. So that's where we got to the last time. And so we opened it up. What is this called? Where it's split open? Starts with an R. It's 2 words. So you definitely want to write this down. You should have been able to read it right off your notes. This is called the replication fork. The replication fork. So before the 2 strands, I'll do it like this since they're, can you guys see the 2 markers? Yeah. They're kind of anti-parallel, right? So when we cut it, when the helicase cuts it, now I have my replication fork. It's not a fork like that has like the 4 things that you stick in, you can eat your food with. It's just a fork like a fork in the road. Are we okay? This is the replication fork, right? I'm talking about if you were going down a path and you had 2 choices, this is called the fork in the road. You have 2 choices on your path. So in this case, we have our piece of DNA, right? Helicase cuts it, and we now have a replication fork. Instead of a path that you would walk on with 2 choices, this is a replication fork. We've got to copy both sides. Does that make sense? Yeah? Okay. Then we talked about adding, so we already added here in this end on the green one, the new complementary strand on this end. What is this end? Is it 5' or 3'? 5'. And so on this end is, on the new strand, this end has to be 3 because you can only have a 5 and then a 3'. DNA polymerase adds to which end? So can DNA polymerase add here? All right, that is adenine that's on the original strand. That's adenine that's on the original strand. So what does Chargaff say needs to be added? The thymine. So go ahead and add your thymine. I'm going to add a thymine. All right, so this is stuff you should, over here on the right-hand side, you should write down. Because you've got to know that sequence. I've added my thymine there. Now, what type of bond forms between the thymine and the adenine?
Hydrogen.
Hydrogen bond is absolutely right. Is that the only bond DNA polymerase has to help form? Or is there another bond? Just based on my question, the answer should be yes, right? Because DNA, remember, it's two strands. Each strand is connected by a hydrogen bond, but to make the strand, you have to have the phosphate of the new nucleotide right here added to this one. What does it add to? What this phosphate adds to this nucleotide, where does it add to? What's the name? Or what carbon is that? It's probably a better way of putting it. The third one, so the number 3 carbon. So far, so good? Okay, so when DNA polymerase adds a nucleotide, how many bonds have to form? So when DNA polymerase adds a nucleotide, how many bonds have to form? 2. This is stuff you have to memorize, and I would definitely write down. When DNA polymerase adds a nucleotide, there's 2 bonds that have to form. 2 bonds that have to form. This is definitely write down worthy stuff. One bond is between the new nucleotide and the old one. What's that bond? One bond is between the new nucleotide and the original. What's the name of that bond between those two? A hydrogen bond, right? The other bond is between the phosphate and the number 3 carbon. It's a covalent bond. I don't think you'll be asked that, but between the phosphate and the number 3 carbon of the next one. Okay?
What about the nucleotide structure? Yeah.
So that same thing had to happen.
But when is the first one?
So part of the honors biology program doesn't mean that there's another enzyme that literally adds a primer on that tells DNA polymerase where to add. Okay, so we don't. That's AP. So we don't do that here. So what we just say is that DNA polymerase adds that first one on.
And you won't ask like how many bonds.
I could ask you again, once it adds this on, this first one, there isn't only one bond forms, right? But this next one, how many bonds would form? 2, and so on, all the way back towards the replication origins, right? Right? Is that okay? Does everybody kind of understand that? Technically, and again, this is beyond the scope of honors bio, just so you know, there's a thing called RNA primase, and it adds primers in a very specific spot as this thing opens up. But that's an enzyme that College Board says honors or pre-AP biology does not need to know yet. In AP bio, that's when you'll get the additional enzymes, okay? Your big focus for this class needs to be that DNA polymerase adds to the 3' end. And it's okay, I get what you're saying, but it's okay. There should be 2 bonds formed. Not the first one, I get that, right? but there should be two bonds formed, one between the original and the new, and that's a hydrogen bond. You've got to memorize that. The other bond that forms is between the new one that was just put on and the one right next to it. Are we okay? All right, can we add another one? Can DNA polymerase add another one? Sure can add another nucleotide. So let's go ahead and do that. And let's add about 3 or 4 more. And just go ahead and add them all the way up there. And when you're adding them, which direction are they be adding? That's good English. Which direction are they adding? Are they adding towards the replication fork, or is it adding away from the replication fork? Towards the replication fork. That is incredibly important. And we'll do it again here in just a minute. So there I've added my next one. I'm going to put my hydrogen bond is together. Now I'm going to put the phosphate sugar bond together. All right, the next one. Going to add the next nucleotide. DNA polymerase 3 adds to the 3' end. I still have a 3' end available. So I'm going to keep going. I'm going to keep adding. Based on Chargaff's rules, guanine always adds to cytosine. So I'm just going to keep going. I'm going to go up a few. It's not overtly critical that you add exactly the same number that I do. In other words, go all the way up to where I go. But you want to go up a few here. So there we go. I'm going to add another one. Just keep adding there as you go up. So this side, are we adding towards the replication fork or away from the replication fork? We're adding towards the replication fork. So I'm going to bend this over a little bit just to make it a little easier. So we're going towards that replication fork. So this strand on this side, I'll wait for just a minute so you can catch up because I know you're putting molecules on it. Yeah, that's probably a good idea to put tape on the back. I see some of you doing that. That's a really good idea so it doesn't stick. Well, I'm going to do the same thing while everybody's putting their stuff together. Yeah, no, it's not going to stick. All right, again, you were adding which direction? Toward or away from the replication fork? Toward. So this is going to be called the leading strand because we're going toward the replication fork. And I'll explain what that means in just a minute. But it's going to be called the leading strand because it's going towards the replication fork. All right. So this is the leading strand because it's going towards that origins of replication, towards that center spot. It'll make sense in just a minute, so don't panic yet. What does he mean? So nucleotides are added I'm going to put origins of replication. And what that means is that spot right there where it started splitting.
Does it always have its own?
The leading strand will always add towards the replication fork. One side will always add towards the replication fork. And we're getting ready to see what the other side is going to do. It'll make sense, I promise. If you give me 3 minutes, it'll make sense. Everybody good so far up to this point? Okay. Just for the sake of clarity, I'm going to kind of pull this over like that, just to make it easier to see. Okay? All right. So let's have a look at this other side. All righty. So first thing that happens is DNA unwinds and helicase opens it up, breaks those hydrogen bonds. I have this side where there was a thymine, so we added adenine. The third prime end is here, so we can go and keep adding onto this end. On this side, the first one is adenine. So what bonds with adenine? Thymine. So I'm going to grab one of my thymines. Now, before you do anything, don't glue anything down yet. I'm going to grab one of my thymines. There shouldn't be anything here at this point. I'm going to grab one of my thymines. There it goes on the floor. I'm just going to put a magnet right on top of it so that we can kind of see what's going on. All right, so that new nucleotide, I could maybe add that, but here's the problem. If I add that there, what end is this end on the new strand that's going to be forming? Is this the 3 or the 5 or the 3? This is 3. This side must then be 5. Okay. Okay, so this end right here is the 3' end, correct? This is the 5' end. So DNA polymerase only can add to the 3' end. So far, so good? So DNA polymerase is going to add to this end, not above it. It's going to add here. I've got another C, so we're going to add a G there. And again, DNA polymerase, when it adds, not the first one, but when it adds, it has to add the hydrogen bond, right? That has to form. And then the phosphate sugar bond here also has to form. That's a covalent bond. You need a name for it. So it's going to add that same piece right here. Okay? Again, helicase breaks those bonds, opens up. One side, immediately we have a 3' end and we have another nucleotide so we can keep adding. On this side, because we cut through it, this side, this other side, because it's the 5' end here, it has to wait. It has to wait until a certain amount gets undone. Usually that's hundreds of nucleotides that helicase has to cut through. And when it's done cutting through those, there's a spot, and again, we talked a little bit about this being there was a primer that was there that has to tell it where to start. You don't have to know any of that for AP. But when it starts adding again to the 3' end only. And so now it's adding away from the replication fork. Do you see this? This side, when it opened, we could immediately add one because there was a 3' end and we can keep going towards that origins of replication. This other side, it had to wait. It couldn't do it immediately. It had to follow. It had to wait. And so at some point up on the strand, it added the nucleotide, and now it has the requirements for DNA polymerase to add. So far, so good? We okay? We okay?
Yeah.
This is called the lagging strand. And so it lags behind. What happens is, again, helicase breaks the 2 strands, breaks them open, So the two strands are broken open. One side can immediately start adding nucleotides because we have to have a 3' end. The other side has to wait. It has to lag behind. Once a certain amount, again, you don't have to know what that amount is. Even AP doesn't. Once a certain amount has been opened, it can then start adding because it has stuff going back down. So let's go ahead and add these here. Okay. So it's going to start. So what goes with G? What's the base that bonds with G? Cytosine. On your manipulative that you have there, it doesn't matter specifically where you start, just as long as you start up above. And again, when you add the nucleotide, DNA polymerase, there's two bonds that form. One bond forms the hydrogen bond between the old and the new, and the other bond is between the phosphate of one nucleotide and the sugar of the next. So I'm going to add it all the way back to the end. All the way back to the end. Okay. All the way back to the end. There we go. And again, please just listen, keep adding. Helicase, the enzyme, we got the unwinding that's happened. We split the hydrogen bonds. One side of the molecule can add immediately because we have our 3' end pointing back towards the replication fork, because we have the 3' end basically pointing back to another nucleotide. The other side has to wait. It has to wait until a certain amount has been split. Till a certain amount has been split, and then it can start adding, because if it had added just down here at the beginning, it wouldn't have gone anywhere. This other side, called the lagging strand, adds away from the origins of replication. It adds in the other direction. It adds away from the origins of replication. Okay? So far, so good. So the lagging strand nucleotides are added away from the origins of replication. And again, there's pieces here, so don't panic. There's pieces here that once everything gets done here in just a minute, we'll go back over it one more time so you kind of understand what's going on. All right, everybody so far okay? Okay. So at this point, we need to open up more of the DNA molecule. So what's the name of the enzyme that breaks the hydrogen bond so that the DNA strands split?
Wait, what was the question? Sorry.
What's the name of the enzyme that breaks those hydrogen bonds so that DNA splits open? Helicase, right. So here we go. I'm going to use my scissors, pretend it's helicase. I'm not going to go all the way through I'm just going to go up enough. Just going to go up enough so we can add more. So I've opened it up more, right? Helicase has opened it up more. Can we begin adding to this side? Can we add to this side, to this leading strand side? Can we start adding right here? Yes, because what end is this? The 3' end. So I can continue to add. So far, so good. I could put a guanine there, because there's a cytosine on the other side. So I can keep adding. Can I keep adding and without stopping all the way back to the origins of replication? Can I keep going? I can keep going, right? I don't have to stop. Once this thing starts opening up more, DNA polymerase can start adding nucleotides, and it can keep going. It doesn't stop. What about the lagging strand side? Can I add one here? I cannot add one here. Why can't I add a cytosine here? Because there's only a 5' on the other side. This is 5' here, and it can't get in there. So what has to happen? This side's going along. What has to happen on this side? Has to wait. And so once it waits, then you can come up and add another. I won't go that high because I need to show you something here real quick. I don't want to run out of time. I think it's adenine is what I need. So I'm going to come up here to one of the thymines on the original strand. So I have to wait to add it up here. Can I then start adding back in? I can then start adding back in. I'm going to go ahead and add those real quick. And I'm just going to do it real quick. So I'll throw in my sugar phosphate bond once I get them all added. All right, so I've added them back in, but I couldn't add anything here. This spot right here, those aren't connected. See, DNA polymerase can't add another nucleotide, nor can it join these fragments. So I have one fragment here, and I have another fragment here. See the two fragments? These fragments are called Okazaki fragments. Those are called Okazaki fragments. It is actually Japanese. I believe it was a husband and wife team, Dr. Okazaki and Dr. Okazaki, are the ones that discovered it in like 60s, something like that. Something I can't remember. All right, so there is one more thing then, because on your leading strand side, we can keep going, right? And eventually we'll have another strand. One strand that's original, one strand that's complementary. But this side, we need to figure out how to join those. So the last thing you need is DNA ligase. And DNA ligase joins the Okazaki fragments. Now, I wouldn't join them together. I would leave them separate so that you could use that to study. So the Okazaki fragments are formed only on the lagging strand. And then what comes in is another enzyme called DNA ligase that joins these. So now we have one complete strand. So all of these things here get put in. I'm going to be a little lazy in the interest of time. We can keep going on this side, right? All the way up. But on this side now, we have to start again here, and so we'll end up having 3 fragments that get put together by DNA ligase. So we started off with two strands that are joined together by hydrogen bonds. And when we made the copy, each copy has one original strand. Everybody okay with that? And each copy has one new strand. This is called semi-conservative replication. It's not that we made a copy and so now we have one copy and another, like when I make a copy of things for you. I have a master, I put it on the computer, on the copy machine, I hit a button, and it gives me copies of that one. In this case, here was my original, but now I have attached to the original one copy, a complementary strand. That's called semi-conservative replication. And then to kind of help explain what you were having before, what question you had before, is it all right if I erase over here? Are we okay if I erase over here?
Yeah.
Okay. So to help answer that question that you had a minute ago, if this is the strand that's being copied, right, and it's happening in multiple places, this is my 3', this is my 5. The 3' end goes towards the replication fork, so that means replication is going that way as a leading strand. Okay? But it's going this way as a lagging strand. But on this side, it's going this direction as a leading strand. And it's going this direction as a lagging strand. So if you were to look at this side, it's going to be different than this side. This is called a replication bubble. Eventually, it all gets copied, and then DNA ligase comes in and fixes all the fragments so that it will all get done. What you don't need to worry about is where it's happening, where it starts and where it ends on a particular piece of DNA. Okay? The textbook piece, the reading I asked you to do, literally has a picture that shows this. Okay, so please make sure that you've done that reading. All right, that's all for